Hi everyone, it's Nick. So I did my project on sickle cell anemia. The reason why I did this is that my background is actually in medical laboratory science, and I'm actually certified to work in a hematology lab. So I chose this topic specifically because I want to know the more genetic molecular basis for this disease. So what is the purpose of this presentation? The purpose is to describe the sickle cell disease, describe the gene responsible for it, outline the molecular cause, describe the mode of inheritance, and even options for genetic testing. Because like I said, my perspective is more in the medical lab where I would see it under the microscope. So what is sickle cell disease? So sickle cell disease is a group of disorders that specifically affect hemoglobin. Hemoglobin is an important protein that's found in red blood cells that is responsible for delivering oxygen to the tissues of your body. So typically, hemoglobin is affected in such a way that deoxyhemoglobin, or hemoglobin without oxygen, appears as a sickle cell under the microscope. So if you see in the backsplash right here, this is what we call a drepanocyte, also known as a sickle cell. This is what I would see under the microscope in the hematology lab. To give you some perspective is that red blood cells, also known as erythrocytes, typically have that circular round shape that almost look like a donut. And even if you want to take a look right here, is that this cell right here, this has a very large center to it, and that's typically what we see in anemia, because there's not that much iron, there's not much more red blood cell space to make that nice pink look that this one right here shows. So before we get in, I want to talk about the sickle cell anemia distribution in the United States. I found that sickle cell is actually more distributed in African Americans and Hispanic Americans than the general population. And as a genetic disease, I thought that this was pretty interesting, as there could be an evolutionary component to this. After further reading, I saw that there's actually a distribution of sickle cell anemia and malaria that's pretty similar. If you look at Africa, in both the sickle cell and the malaria distribution, is that they're both pretty heavily populated. Same thing with South America in both of them. This is because sickle cell actually resists Plasmodium, which is the parasite that's responsible for malaria. Because sickle cells are in such a way that they are smaller, Plasmodium has a harder time getting in and replicating itself. So I thought that was pretty interesting. So what are some symptoms of sickle cell anemia? Well, first of all, sickle cells are fragile, and they tend to lyse easier or break apart. And this is what really causes that anemia, is that breaking of that red blood cells. And anemia can cause shortness of breath, general fatigue, and other symptoms. Because again, without your tissues getting oxygen, you'll start to feel like you need more oxygen in your body. Another thing that sickle cells can do is something called a vaso-occlusive crisis. And this is, if you look at the picture right here, is when they occlude a vein to the point where it impedes other erythrocytes from going through. This can cause an immense amount of pain due to ischemia or lack of oxygen, and this can actually cause end organ damage. Another thing that's pretty interesting is that an important organ in your body called the spleen is responsible for culling abnormal red blood cells from your circulatory system, and these sickle cells can actually block up the spleen and cause what we call splenomegaly, which is an enlargement of the spleen. And sometimes you actually need that to be removed in order for the body to recover from that enlarged spleen. So what specifically causes sickle cell disease? So sickle cell disease is a genetic disease, as we said, that is characterized by a mutation of the HBB gene, which is responsible for coding beta chains that make up hemoglobin. Because like I said before, hemoglobin is responsible for delivering oxygen. Hemoglobin is made up of two different chains, both alpha and beta chains. Sickle cell disease specifically affects the beta chains in hemoglobin. So this gene is located on chromosome 11 at position 15.4. Hemoglobin S, the gene that causes sickle cell disease, replaces that normal beta globin subunits. So what is the molecular cause? So if you see on the left picture right here, we have glutamate. This is a normal hemoglobin that can go from deoxyhemoglobin to oxyhemoglobin in a very easy, normal fashion. Unlike, as we see right here, this valine. This valine is really what's responsible for giving those drepanocytes or sickle cells that sickle cell shape. Is that this valine in deoxyhemoglobin creates that, again, that sickle cell shape. And this is caused by, again, valine replaces glutamic acid. Not only that, but we can also see right here is these are the heme units. There are 4 heme units in hemoglobin, and that's why hemoglobin can carry up to 4 oxygen molecules. But since sickle cell is an oddly shaped cell, is that hemoglobin can only carry 2 oxygen in a sickle cell. So what is the mode of inheritance for sickle cell disease? Well, sickle cell has an autosomal recessive mode of inheritance. What this means is that there's no sex determinant of this disease, meaning that both sexes get affected equally, and that if it's a recessive, that a person needs both copies of the sickle cell gene in order to express this trait. Excuse me. So if a person only inherits one copy of the sickle cell disease, they are considered a carrier and may exhibit a sickle cell trait. So this is a pedigree of sickle cell anemia, and I think that this is a really nice way to really see how sickle cell gets inherited down the line. So if we see here, Catherine is a sickle cell, she's afflicted by sickle cell disease, while John is a carrier. And if you see by their offspring, every single one of them is either a carrier or they're afflicted. And we can easily do a Punnett square real fast to see why that is. So we have John right here. Ooh, excuse me. So we have John right here. And we also have Catherine. Ooh. Oh, sorry, it's not letting me right up there. So if you see right here, there's a 50-50 chance that they're either going to have sickle cell disease or they're going to be a carrier of it. And what I found pretty interesting is that if we see down here with Bruce and Martha, is that when a sickle cell afflicted person copulates with a non-affected person, every single one of them is going to be a carrier. So if we look right here, here's Bruce. And here's Martha. As you can see right here, it's a 100% chance that all their offspring will be a carrier of it without necessarily expressing fully a sickle cell disease patient. And I think that this is why sickle cell can easily go through families without people really noticing it, because again, only it takes one person to be afflicted by it to create so many carriers, which will then pass down those genes. So there's some genetic testing available, like microsatellite instability testing, deletion and duplication analysis, sequence analysis of select exons, and targeted variant analysis. These are all good tests to see if you have this aberrant gene, but typically the testing can either be done in the clinical lab, like putting it under a microscope, or we can do a saline test where we put your sickle cells in a test tube. So here's a test tube and we put your sickle cells in there, and then we just add saline, and then the turbidity of the solution will be pretty great with those cells lysing. Or we could also do like high performance liquid chromatography just to see the retention rates, because the retention rates between sickle cell anemia and a regular hemoglobin differ. So like I said, genetic testing is available, but typically it could be cost prohibitive just because, again, these are molecular tests. They usually cost a little bit more. They require either a genetic counselor or someone specifically certified in genetic testing to do these. But these are available for phylogeny testing, or just in case of people really want to know what type of sickle cell they may have and what type of gene may be affected. Other than that, this is my bibliography. I do appreciate you guys coming out and seeing my presentation. I had a fun time recording and making it. If you have any questions for me, you can either reach me by email or just shout me out in the forums. So I look forward to all your presentations, and I hope you guys have a good rest of your summer.